Welcome to B004. Today we explore DNA, the molecule of heredity. In 1953, James Watson and Francis Crick published the double helix structure of DNA, arguably the most important scientific discovery of the 20th century. This structure immediately suggested how genetic information could be copied and transmitted. Today we will understand the architecture of DNA, how it is replicated with extraordinary accuracy, and what happens when that accuracy fails, mutations. This is foundational to genetics, molecular biology, and medicine. Hook, your genome is 2 meters long, in every cell. Consider this: the DNA in a single human cell, if stretched out in a line, would measure approximately 2 meters. You have approximately 37 trillion cells. That means if you laid out all your DNA end to end, it would stretch around 70 trillion meters, enough to go from Earth to Pluto and back more than 17,000 times. Yet this 2 meters of DNA fits inside a nucleus that is only 6 micrometers across. It does this by coiling around proteins called histones, creating nucleosomes, which then coil further into chromatin fibers, and further still into the densely packed chromosomes visible during cell division. Understanding this packaging is crucial because the tightness of packaging directly controls whether genes can be read or not. DNA is a double helix composed of 2 antiparallel polynucleotide strands. Each nucleotide has 3 parts: a phosphate group, a deoxyribose sugar, and one of 4 nitrogenous bases. The bases are adenine and guanine purines with 2 rings, and thymine and cytosine pyrimidines with 1 ring. Chargaff's rule is critical: adenine always pairs with thymine via 2 hydrogen bonds, and guanine always pairs with cytosine via 3 hydrogen bonds. This strict pairing means the percentage of adenine always equals the percentage of thymine, and percentage guanine always equals percentage cytosine. The 2 strands run antiparallel, one goes 5' to 3' while the other goes 3' to 5'. The 3 hydrogen bonds in GC pairs make GC-rich DNA slightly more stable and harder to denature. DNA replication is semi-conservative. Each daughter molecule gets 1 original strand and 1 newly made strand. This was proved by the Michelson-Stahl experiment in 1958 using nitrogen isotopes. Replication begins at origins of replication and proceeds bidirectionally. Helicase unwinds the double helix, creating a replication fork. Primase lays down RNA primers because DNA polymerase cannot start a new chain, it can only extend. DNA polymerase III then adds deoxyribonucleotides, but only in the 5' to 3' direction. This creates an asymmetry: the leading strand is synthesized continuously toward the fork, but the lagging strand must be synthesized in short Okazaki fragments moving away from the fork. DNA polymerase I replaces the RNA primers with DNA, and ligase seals all the fragments together. The entire human genome is replicated in about 8 hours, with only 1 error per billion base pairs. Mutations are permanent changes in the DNA sequence. Point mutations change 1 nucleotide. A silent mutation changes the codon but codes for the same amino acid due to the degeneracy of the genetic code, so the protein is unchanged. A missense mutation results in a different amino acid, potentially changing protein function. Sickle cell disease results from a single missense mutation changing glutamate to valine in the 6th position of the hemoglobin beta chain, causing the protein to polymerize and distort red blood cells. A nonsense mutation creates a premature stop codon, truncating the protein. Frameshift mutations are usually the most severe. Inserting or deleting 1 or 2 nucleotides shifts the entire reading frame, changing every codon from that point to the end of the gene. Cystic fibrosis involves a 3-base deletion, not a frameshift since it is a multiple of 3, but it removes a critical phenylalanine from the CFTR protein. The key facts to lock in for B004. Adenine pairs with thymine via 2 hydrogen bonds, guanine pairs with cytosine via 3. Chargaff's rule means the sum of purines always equals the sum of pyrimidines in double-stranded DNA. DNA polymerase synthesizes only in the 5' to 3' direction. This is non-negotiable in the enzyme's mechanism and explains why the lagging strand needs Okazaki fragments. Replication is semi-conservative, demonstrated decisively by Michelson and Stahl using heavy nitrogen labeling. Mutation severity generally increases from silent through missense, nonsense, to frameshift. Understand the mechanisms of each type because NET frequently presents you with a DNA sequence and asks you to identify the type of mutation and predict its effect on the protein. NET strategy for DNA. Base pairing and Chargaff's rule are used in calculation questions. If you are told a DNA is 30% adenine, you immediately know it is 30% thymine, and therefore 40% combined guanine plus cytosine, which means 20% guanine and 20% cytosine. Practice these calculations until they are automatic. For replication enzyme questions, remember helicase unwinds, primase makes the primer, polymerase III extends, polymerase I replaces primers, and ligase seals. Students often forget ligase in the sequence. For mutation type questions, a deletion or insertion of 3 bases, or any multiple of 3, is not a frameshift, it just removes or adds 1 amino acid. A frameshift requires a non-multiple of 3. 5 takeaways from B004. 1. DNA is a double helix with antiparallel strands, A pairs with T via 2 hydrogen bonds, G pairs with C via 3. 2. Chargaff's rule, %A equals %T, %G equals %C, is used in calculation questions. 3. Replication is semi-conservative, proved by Missense and Stahl. 4. DNA polymerase synthesizes only in the 5' to 3' direction, creating leading and lagging strand asymmetry. 5. Mutation severity increases from silent to missense to nonsense to frameshift. In B005 we follow the information from DNA into protein transcription and translation, the central dogma of molecular biology. See you there.